Welcome to the Blueprint of Life video 17. In this video, we're going to be looking at the work of Beadle and Tatum and how their experiments allowed us to understand that for one gene, there is one polypeptide produced. So in 1941, two scientists, Beadle and Tatum, started working on bread mold or a fungus called Neurospora crassa. And this led them to the idea that genes provided the instructions for making proteins, which is what we accept today. When they tested this mold, they grew it on a minimal medium, which means that it had only the things needed for the mold to grow, but it had no extra amino acids added. And we can see here that this mold that was produced was then exposed to some UV light and X-rays, and then they grew it again, this time on a complete medium. They selected one of the spores, grew it on a complete medium, and then they saw that on when that spore was selected after it had been exposed to UV radiation, when it was grown on a minimal medium, it didn't grow, which led rise to the idea that there was a gene that was damaged from the UV and the X-rays that were that it was treated to, and that it stopped growing an enzyme which was responsible for these different things that allowed the mold to grow. And then we see here that they grew it on minimal media with different supplements in it, and you can see that it all of them grew. All of them didn't grow except for the one with arginine, so that they found that the arginine enzyme was in fact damaged from this radiation. This led them to produce the one gene, one enzyme hypothesis. So they induced these mutations. They couldn't produce the amino acid, which allowed them to grow, which they thought was due to a particular enzyme not functioning. Then, when they did a few more crosses, they showed that the inability to produce this amino acid was in fact inherited, so it was passed down through the generations. Different enzymes in some of the different mutants were altered or missing, so then they proposed that a gene determines the structure and function of an enzyme. But then more research came to light, and we now know, and the hypothesis which we follow today is one gene, one protein, or one gene, one polypeptide. Because it was demonstrated that proteins other than enzymes are encoded by genes. They found that it is not, in fact, one gene responsible for the structure of an entire protein, but that protein is, in fact, made up of many polypeptide chains. So that means that this hypothesis, we now know, the one gene, one enzyme was incorrect, and we now follow the one gene, one polypeptide. This was also because not all proteins are enzymes. So that is the work of Beadle and Tatum. Make sure you tune in for video 18, and make sure you do the secondary research task that we have to do in class to further summarize the evidence and understand why the hypothesis was altered.